Hello everyone, this is MMBS111 Introductory Microbiology. This is mini lecture 8.4, DNA recombination events. There are 5 learning outcomes for this mini lecture. First, define recombination and recombinant. Second, describe horizontal gene transfer and the 3 types in bacteria. Third, explain the role of a competent cell in transformation. 4, name the carrier in transduction, and 5, define transposons. So we need to define a couple of terms, recombination and recombinant. When we're talking about recombination, we're talking about how the genetic information of an organism, particularly a bacterium, has recombined or changed. So one bacterium may donate a plasmid to another bacterium, and this results in a new strain that is different from the donor cell as well as the original cell. This often is how genes for drug resistance, toxins, metabolic poisons are transferred. It also tends to increase the virulence or the ability for an organism to cause disease. Recombinant is any organism that contains genes that originated in another organism. So those are the differences there. In bacterium, the ability to recombine their DNA is significantly easier than it is in eukaryotes. This is what we call horizontal gene transfer, where DNA is transferred between organisms that are not parent and offspring. And this can happen through the transfer of plasmids that are able to replicate themselves independently of the cell, or chromosomal fragments that get picked up and integrated into the bacteria's chromosome and then are replicated with that chromosome. There are 3 types of horizontal gene transfer in bacteria that we're going to talk about. And the first is conjugation. And we refer to this kind of as gene sharing. A plasmid, or sometimes some other genetic material in the cell, is transferred to another cell via a direct connection. So the 2 cells are connected. Typically this happens with a pilus. Conjugation is a conservative process because the donor cell retains or conserves a copy of the genetic material that it is transferring to the new cell or the recipient cell. In gram-negative organisms, conjugation involves a pilus to connect the 2 cells and a fertility factor or a plasmid. Okay, and this organism then is referred to as being F positive. It has a plasmid that it can donate or transfer to another cell. In gram-positive organisms, conjugation happens when an opening in the cell wall occurs between 2 very closely adjacent cells. Resistance factors are typically transferred in this way. So this is how organisms acquire antibiotic resistance, is generally through a conjugation event where they get a plasmid that has this resistance gene. Also resistance to heavy metals, and the ability to produce virulence factors like toxins, certain enzymes, and adhesion molecules that allow the organism to be better at causing disease or infection. The next type of horizontal gene transfer is called transformation, and this is what some scientists refer to as the funeral grab. So we have a dead cell, a lysed bacterium that has released its digested or broken down DNA into the environment. And another cell in close proximity happens to be in that same environment nearby, recognizes that DNA and brings it into the cell, incorporates it as part of its chromosome, and then that DNA fragment gets replicated with that organism's normal chromosome. Cells that are able to do this are said to be competent. They are capable of accepting genetic material from the environment. Transfection is a similar process that happens in eukaryotic cells, and this is how we have in the laboratory genetically modified some of these cells, like yeast and plants, some mice cells, through this process. The final form of horizontal gene transfer is called transduction, and this process requires a bacteriophage. There are two types of transduction, generalized and specialized, and each one involves a different type of phage, and a sort of a different event is taking place. So in generalized transduction, a DNA fragment is transferred from one bacterium to another by a lytic bacteriophage due to a mistake during viral assembly. So remember, a lytic bacteriophage is going to infect a bacterium. It's going to stimulate the production of more phages. It's going to lyse the cell so that all of those phages then can be released into the environment to infect new cells. During this process, some of the bacterial DNA gets chopped up or digested in the process of forming the viral DNA, and a piece of that bacterial DNA gets incorporated in the capsid of the phage instead of the viral DNA. And then that bacterial fragment then gets transferred to new cells. So that's generalized transduction. Specialized transduction occurs when a DNA fragment is transferred from one bacterium to another by a temperate bacteriophage due to a mistake during induction. So remember, a temperate bacteriophage is a bacteriophage that infects a bacterium but can either go through the lytic cycle or on more occasions, it becomes dormant and it incorporates its DNA into the chromosome of the bacterium, and it remains dormant for months or years. And that phage DNA is continually replicated by the bacterium as it divides. Remember that certain things can induce a phage to remove itself from the bacterial chromosome and then begin the lytic cycle. So during induction, this temperate phage may take some of the bacterial DNA with it as it removes itself or excises itself from the bacterial chromosome, and then that piece of bacterial DNA gets replicated as part of the lytic cycle and is incorporated with the viral DNA into the capsid, and then gets transferred to other bacterium as the phages infect new organisms. So those are all examples of horizontal gene transfer. Transposons are not exactly horizontal gene transfer, but they do result in the recombination of DNA. Transposons are also known as jumping genes, and they were first discovered by Barbara McClintock when she was studying maize. and noticed the color changes in corn kernels. So these transposable elements, or these jumping genes, can shift from one part of the genome to another. So they can move places or locations within the chromosome. Some of them actually replicate before they move to the next location. So we get a number of copies of that transposon within the chromosome, and others just move to new locations without copying themselves. Sometimes a transposon can move from the chromosome to a plasmid. And when the transposon ends up on the plasmid, then that plasmid can be replicated and it can be transferred through a conjugation event to another bacterium. Transposons are involved in changes in traits such as colony morphology, so what the colony looks like under the microscope, its appearance. It can also result in changes in pigmentation, like what's seen in maize, and antigenic characteristics. It can also be involved in the transfer of drug resistance in bacteria. So that is the end of mini lecture 8.4. Here are a few questions to check in to see how much you understand from this section. Make sure that you go through the questions and that you can respond to them properly. Here's our critical thinking question. So a methicillin-resistant Staphylococcus aureus, or MRSA, outbreak has occurred in a local elderly care facility. 2 separate strains have been isolated from patients and staff in the facility. However, both strains possess the exact same gene sequence for methicillin resistance. Explain one possibility for how this occurred. So here is an example of a question that you might see on an exam. How would you respond to this question, knowing what you know now about horizontal gene transfer and genetic recombination? Make sure that you can go through this question and respond appropriately. Remember that you can watch the videos multiple times. Also refer back to the learning objectives because that's where your exam questions will be pulled from, and make sure you understand those components. And hopefully this video was helpful.